Hello friends, I am Shubhangi Rohatgi, PGT Biology, KV1 AFS Indian, and I welcome you all to the first session of Chapter 11, Transport in Plants, that is for class 11th. Have you ever wondered that how do plants move different things in their body to such large distances? You must have noticed that generally, most plants are way bigger than the animal bodies, yet they do not have any circulatory system. So, how do you think materials are transported inside their bodies? Have you ever heard of any heart transporting things inside a plant? Of course not. So, let us see how do plants carry out such long distance transports inside the plant, and as a matter of fact even how the cell to cell movement of different substances is carried out in the cell. So, let us have a look on the things which are transported inside the cells and the plant body. So, the first and foremost thing that is transported is the water. Water is required for photosynthesis and all the chemical reactions that are occurring in the plant body, and it is to be taken from roots all the ways to the stems and the leaves as well. Then there are mineral nutrients which are also absorbed from the soil and are to be taken to different parts of the body. Then the organic nutrients or the photosynthetes which are produced in the leaves, and then they are to be taken to all the growing parts and even for the storage. And finally, the plant growth regulators or other such chemicals which control or regulate various growth related activities of the plant body. They are produced at one place and then they are needed to be transported to their site of action. So, these and many other similar things are transported in the plant bodies. What are the means of transport? That is, how are they transported? The first and foremost is diffusion. Diffusion is very common inside the plant bodies. It is the random movement of molecules from their high concentration to low concentration, and it is usually used to carry molecules over short distances like gaseous exchanges through stomata. Then there is cytoplasmic streaming. Cytoplasmic streaming is a mechanism which is dependent on ATP, that is, it is an active mode of transport, and in it the cytoplasm moves around in the cell so that the nutrients and other materials are taken to every nook and corner of the cell. Then of course there is active transport, whereby energy in the form of ATP is used to carry out the transport of substances. And finally, there is translocation. Translocation is movement of substances and material over long distances. So, we can classify the means of transport as follows: passive and active transport. In passive transport, energy is not required, but in active transport, energy is used in the form of ATP. When we talk of passive transport, we can study the diffusion, facilitated diffusion and osmosis. All these processes do not require any energy in form of ATP to carry out transportation, or we can say that even diffusion, it is not dependent on living things also. It can be shown even in non-living systems also. Let us talk about the direction of transport, where the transportation is taking place in the plant body, whether it is from roots to leaves, leaves to stems or other organs. Let us study them one by one. First of all, let us take the example of xylem transport, where water and minerals are being transported. Obviously, since the water is to be absorbed from the soil, so the transport in xylem will always be unidirectional from roots to the stems. Water will be taken up, absorbed from the roots, and it will be taken to different parts of the body. So, xylem transport will always be unidirectional. But the same cannot be said of the phloem transport, where organic nutrients are being transported. Look what happens. Suppose this is a plant. Now, as the photosynthesis was taking place somewhere here. So, when the food was made, it was to be transported to all the growing parts where wherever there are stems, buds, growing flowers, the food will be taken to all directions. However, it will also be taken to the roots. They need the food for growth and for storage also. So, the leaves are the sink organs, the leaves are the source organs where the food is being produced, and the storage organs are the sink organs where this food will be stored. Now, so the phloem transport is in this direction when the food has been made. What will happen when there is leaf shedding in autumn, when there are no leaves to produce food? Now, the storage organ, it will act as the source, and food will be taken from the storage organ to the other parts of the plant body, which will now act as sink. So, here we can see that the phloem transport can be multidirectional. Either the leaves or the roots both can act as the source for initiating the phloem transport. Similarly, when we talk about the mineral nutrients, what happens? Minerals were absorbed from the roots along with the water, and then they were taken to the other parts of the plant body. But when the time is for senescence, when the organ is preparing for the senescence, what will happen? Then the minerals are not generally wasted with the senescent organ. Rather, they are transported to other parts of the body. So, here now the mineral nutrients that were stored in the organ which is to be dead, it will remove the nutrients from its sap, and now the transport will be in other direction. So, phloem transport is always multidirectional. Then the hormones or plant growth regulators, they are mostly transported in polarized or unidirectional pathway. Why? Because the site where they are produced is generally different from the site of their action. Their target site is different from the site of synthesis. So, they are always transported in unidirectional pathway. Let us talk about the means of transport. Plant cells, like all other living cells, are surrounded by a semi-permeable membrane, and any particle moving into or out of the cell must cross this membrane. So, there are three different ways by which this membrane can be crossed: diffusion, facilitated diffusion, and active transport. Let us study the diffusion first. Before going into the details of what is diffusion and how does it occur, let us perform a very small activity. You must have performed similar activity in your younger classes, and you must have observed diffusion also. So, let us make it a recall activity. So, you can see we have taken two beakers here, and there is water in these beakers, and the water is quite stable. We can see that superficially nothing is moving around in the beakers, or is it? Let us see. So, what am I going to do is, I will add just a drop of this stain. This is saffron stain. I will add just a drop of the stain in one of the beakers, and we will quietly observe what happens. So, there goes first drop and yet another one to follow. So, the system is apparently not that still. There are movements taking place. If the color is not very clear, let me add a few more drops. Please observe the motion of the stain particles in the system of the water. So, there was liquid and the stain was also liquid, and it has started moving around in the beaker. What if there were some solids? Will solids also follow any such movement? Let us see. I am going to add just a little bit of potassium permanganate in this other beaker. Okay. So, there go the particles of the potassium permanganate. Oh, even the solids are not so still in this system, and we can see them moving around. The movement, though, appears to be random in all the directions. We can see that here and here also the movement is in all the directions, but yet somehow there is one portion where the color is concentrated. In this beaker it is above the water, and in this beaker it is near the bottom of the beaker. So, the color is concentrated at one point and it is seeming to be radiating to all the other parts of the beaker. What is happening? The diffusion is happening. Let us study about diffusion in detail now. Meanwhile, we will let the beakers sit for some time and we will come back to them again to see what has happened after some time has passed. Okay? So, talking about diffusion. According to the kinetic theory, the molecules or ions of all substances are in constant motion in all directions due to their kinetic activity. So, all the substances have kinetic energy, and because of this kinetic energy, they keep on moving. This movement is random, but the overall movement is from a region of high to low concentration, and this is called diffusion. So, what is diffusion? Diffusion is the random movement of particles because of their kinetic energy from the region of their high concentration to the region of their low concentration. And diffusion is the process in which material spreads throughout the liquid or a gas. We will see in a while that the color in the beakers will be spread evenly. Diffusion is a very slow process, and as I had said earlier, it is not dependent on any living system. Diffusion is obvious in gases and liquids. Have you ever smelled the food being cooked far away and that smell has made you hungry? How did those smell particles come to your nose? By diffusion only. Similarly, diffusion in liquids, we have just shown in this activity that diffusion in liquids is also fairly easy. However, in solids, diffusion is somewhat unlikely, but yet it is there, and we have seen that the crystals of potassium permanganate, they diffuse very quickly inside the water. Diffusion is very important for the plants. It performs many activities. Plants transport a host of things with the help of diffusion. Firstly, passing of water and ions of simple organic salts into plants through the root cells, it occurs by diffusion. Then loss of water vapor from leaves and other aerial organs, that is by transpiration. It is also because of diffusion. Then oxygen evolved in photosynthesis and respiratory carbon dioxide. These gases also diffuse out of the plant into the atmosphere with the help of diffusion only. Then the movement of substances within a plant, that is from one part of the plant to another, is mainly by diffusion. The gases move through the intercellular spaces and water and solutes through the dead and living vessels, and also from cell to cell by diffusion. So, we can sum up to say that almost all the short distance transports in the plants are carried over by diffusion, whether it be of gaseous molecules or the liquids. Most of the short distance transports are carried over by the process of diffusion without spending any energy in the form of ATP. We have to keep this in mind that diffusion will always take place from the region of high concentration of one substance to the region of low concentration of that very substance, from high to low. this is what I was talking about, that direction of diffusion is independent of the direction of diffusion of other ions or molecules. Suppose 2 or 3 ions or molecules are being diffused. So, direction of diffusion of one molecule or ion will be totally independent of direction of movement of other ion or molecule. It will depend only on its own concentration. For example, the outward diffusing water vapor molecules in transpiration, they do not carry respiratory carbon dioxide with them. It does not happen that if the water molecules are going out, they will take the carbon dioxide along with themselves. No, but each diffuses independently of other, depending only on the differences of its own concentration or pressure inside and outside the plant. Let us talk about the rates of diffusion. Rates of diffusions are dependent on concentration gradient, temperature, pressure and density, and also the permeability of membrane. What do we mean by permeability of membrane? It is the ability of a membrane to let the substances pass through them or not. It can control whether it wants a material to pass through it or not, and this is the permeability. We all know that the membranes in our systems are semi-permeable, that is, they allow the free movement of some particles but restrict the movements of others. This is the diagram of fluid mosaic model of plasma membrane given in your NCERT books, and here we can see that the plasma membrane is made up of lipid bilayer and many different proteins are spanning across the length and width of the plasma membrane, and these proteins are integral proteins and they are transmembrane proteins. They are passing from one side of the membrane to other side. So, before studying the transport in detail, let us have a very brief discussion on the structure of the cell membranes. Cell membranes are made up of bilipid layers. A cartoon of lipid molecule is drawn somewhat like this. So, this is the glycerol head along with the phosphate group. This phosphate group present here makes it negatively charged and hydrophilic. The fatty acid chains that are present here they are hydrophobic. So, what will happen? These fatty acid chains are nonpolar and they are also hydrophobic. So, what will happen that whenever such a lipid molecule is present in a system, it will arrange itself in such a manner that only the hydrophilic heads are towards the solution and water hating, water afraiding hydrophobic tails, they will be towards inside. So, if there are two layers of lipid molecules, they will be arranged in this manner. All the hydrophobic tails will be towards inside and hydrophilic heads will be towards outside. So, what happens that the transport of materials across this membrane, it is dependent upon the nature of these hydrophilic and hydrophobic parts. Let us see how. The water molecules, which are very small, they can penetrate the lipid bilayer by passing between the phospholipids. Just because they are very small, they simply pass by penetrating it. Similarly, few other hydrophilic molecules such as methanol and ethanol, they can also penetrate the hydrophobic membrane because of their small size and lack of charge. Though they are hydrophilic, but yet they can pass through this hydrophobic part because of their small size and lack of charge. And what about the hydrophobic substances? Well, they have no difficulty. Like oxygen, they can cross the membrane very easily because they are lipid soluble and they simply dissolve into the membrane through to the other side. But the problem comes when there are large molecules and they need to be transported. How will they transport? They cannot pass the lipid bilayer by simple diffusion. If there are large molecules, large hydrophilic molecules which have to pass through this area, hydrophobic area, it will be very difficult for them to pass through this area, and so they cannot pass through simple diffusion. They can cross the membrane only with the assistance from specialized transport proteins known as carrier proteins. They are also known as permeases, and such a diffusion with the help of carrier proteins is called as facilitated diffusion. The facilitated diffusion is the movement of specific molecules down a concentration gradient and via specific carrier protein. So, we must remember that facilitated diffusion is also a type of diffusion only, because here no energy in the form of ATP is being used, and here also the movement is from high concentration to low concentration only. The only difference is that the carrier proteins or the permeases are involved. They aid the movement of these hydrophilic large substances through the lipid bilayer. Each carrier has its own shape and it is very specific. The selection is by size, shape and the charge. Examples of facilitated diffusion, like passing of potassium ions through a membrane with an potassium transport protein and passing of glucose and amino acids with the aid of proteins called permeases. Similarly, there is a retinol binding protein also which helps to transport retinol and fatty acids. How do these proteins help in transport of substances? They may change their shape to let a passing molecule through. Many transport proteins appear to work by a shuttle type of mechanism, binding to recognized substances on one side of the membrane and then releasing them on the other side. Other carrier proteins such as retinol binding proteins, they do not change. So, what do these carrier proteins do? They bind to the substance to be transported on one side and then they change their shape and or change their direction, and in this manner this substance which was to be transported is taken to the other side of the membrane. Under facilitated diffusion, we can study three types of transports. Uniport, when a particular type of molecule moves through a membrane with the help of carrier protein. So, this substance A is being transported to other side with the help of carrier protein. This is uniport. Symport is when two kinds of molecules move in same direction while diffusing through carrier proteins, and this two solutes transport is obligatorily coupled. That is, a gradient of one substance, usually an ion, may drive uphill transport of a co-substrate, and it is sometimes referred to as secondary active transport also. So, here the two substances are being transported in one direction with the help of one carrier proteins. So, this is symport. Antiport is also movement of or transport of two substances with the help of one carrier protein, but the only difference is that here the direction of the transport is totally opposite. So, they seem to be getting exchanged. Talking about the carrier proteins, these proteins as I had said earlier that they span the plasma membrane. They are transmembrane proteins. So, they run from one side of the membrane to the other side, and they control the response of cells to certain growth factors and hormones whose binding to the cell membrane causes channels for facilitated diffusion to open or close selectively. Transport proteins are tailored to let only particular molecules through, that is, they are very specific, and because of this specificity the membranes become permeable, semi-permeable. There are some special transport proteins like porins. These are barrel proteins and they act as support for molecules to diffuse. They are quite large and they can allow the passive diffusion, but again they are very specific and they are usually made for medium sized or charged molecules like sugars, ions and amino acids. They are generally present in outer membrane of gram negative bacteria, some gram positive bacteria of the group Mycolata and mitochondria and chloroplast. Aquaporins are also special channels. They are used for transport of water. They selectively conduct water in and out of the cell, but they do not allow the solutes to pass through them. So, they are the plumbing systems of the cell. Every cell is primarily water, but the water does not just sit in the cell, it moves through it in a very organized way, and this process occurs rapidly in the tissues that have aquaporins. As I had said earlier, that the presence of these carrier proteins make the membrane selectively permeable, and the nature of the membrane being selectively permeable is very important. Without membranes, separate compartments could not be maintained to separate the various incompatible biochemical reactions of living systems. And without the semi-permeability, the biological environment within the various compartments could not be continuously replenished and would be unable to respond to a continuously changing environment. This was about the passive transport, where energy was not used for transport of substances. But since the energy was not used, so we think that it is good, but the problem with the passive transport is this: it will always transport molecules from the region of high concentration to their region of low concentration. What about instances when we need to collect ions or molecules against their concentration gradient? That is, if we want to collect some ions or molecules on one side of membrane where already the concentration is very high, from the other side where is their concentration is low, at that time ATP needs to be used, and the process is active transport. Active transport requires a transmembrane protein called a transporter and energy, that is the ATP. Unlike passive transport, which uses kinetic energy and natural entropy of molecules moving down a gradient, active transport uses cellular energy to move them against the gradient, polar repulsion or other resistance. So, in active transport, the membrane proteins, they take the molecules from their low concentration to high concentration, and the work must be done to overcome the influences of diffusion and osmosis. Active transport can be of two types: direct or primary, or indirect or secondary. When the ATP are used directly, it is known as direct or primary active transport. For example, in sodium potassium pump, here the energy is used directly and it is obtained from the hydrolysis of ATP. In indirect or secondary active transport, what happens that the energy of already stored gradient is used for transport of another material. Direct active transport of the ion establishes a concentration gradient. When this is relieved by facilitated diffusion, the energy released can be harnessed to the pumping of some other ion or molecule. Example, glucose and sodium transport. Sodium wants to get inside, and energy released by its transfer will be used to transport the glucose. Similarly, in mitochondria and chloroplast also, the electron transport chain where ATP formation is taking place, the proton gradient of produced by continuous transport of protons, it powers the phosphorylation of ATP associated with ATP synthase. So, let us compare the different mechanisms that we have studied: simple diffusion, facilitated transport and active transport. As we can see that diffusion does not need special membrane proteins, but facilitate transport and active transports, they need the membrane proteins, and because of the presence of membrane proteins, they are highly selective because the proteins are specific in their functions, and the transport can saturate also. When all the protein molecules along the membrane are occupied, the rate of transport will become saturated. Uphill transport means transport against the concentration gradient. It cannot take place in diffusion or facilitated transport, and it can only take place in active transport, and that is why it requires energy. Let us move back to our beakers. Let us see what has happened. So, it has been about 20 minutes since we added the drops of safranin and potassium permanganate in these beakers. What has happened? No, nobody has touched these beakers, and we can see that the particles of the solutes, they are trying to get even. That is, a process is being done to reach the equilibrium. Now, if we leave these beakers in this same state for more time, we will see that the particles of these stains or solutes will be evenly distributed all through the beaker. So, we will get an even solution. This is the movement towards that even solution only. This was about diffusion and active transport. Let us move on to next topic, plant water relations. Water is very important in plants for their day-to-day functions. Structurally, we know that water makes up to 80 to 90% of fresh weight of herbaceous plants and more than 50% of woody species. Also, in it, the proteins and fats form the protoplasm. It also fills the vacuoles which permit it to function accordingly. Then, cell expansion is also caused by retention of water inside the cell, and water acts as a solvent for organic and mineral nutrients as well as gases, and it aids in transport of these molecules. This is not all. Even photosynthesis and other hydrolysis reactions, they are also dependent on water. It supplies the electrons and the protons needed for light reaction. Though the amount involved is small, yet it is critical. Also, just as in our bodies water helps us to keep us cool, similarly in plants also water helps to cool them. Talking about water potential. See this picture. This is one of the tallest trees present on the face of the earth, the sequoia, and here what is happening? The pressure from the roots of the tree seem to have broken the pavement which is made up of cement. How is it possible? So, plants can move water to the top of a 116-meter tall tree. How? Plants can also generate enough force to split rocks and buckle sidewalks, as we can see in this picture. The question is, how is it possible? Well, they use only the basic laws of physics and the simple manipulation of potential energy and hydraulics to carry out these functions. Let us see what happens. Actually, water potential is a measure of ability of water molecules to move freely in the solution. That is, water potential is the measure of its kinetic energy. Water molecules possess kinetic energy, and in liquid and gaseous form they are always in random motion. We had just seen that also in case of diffusion. So, the greater the concentration of water in a system, obviously the greater will be its kinetic energy or water potential. That means the pure water has highest water potential, as there is no solute, all of the water molecules are free to move. These points are very important to remember. Firstly, that water potential is the measure of kinetic energy, the free energy available, and the pure water has highest water potential because the solute molecules are not there to hinder the random movement or flow of the water molecules. But when we add a solute, what happens? It is attracted to water molecules. So, these water molecules can no longer freely move and the water potential is lowered. So, another point to remember is that whenever you add solute to water system, its potential will be lowered. Since the pure water has highest potential, whenever a solute will be added to it, its potential will be lowered. If two systems containing water are in contact, random movement of water molecules will result in net movement of water molecules. But what will be the direction of this movement? From high water potential to low water potential. This process of movement of substances is obviously the diffusion. The energy required per quantity of water to transport water from a sample to reference pool of pure free water is also the water potential. Suppose you have water in soil sample and there is water in a drinking glass. The water in the glass is relatively free and available, but the water in soil is bound to surfaces, diluted by solutes and under pressure or tension. So, the free water can be accessed without exerting any energy. The soil water can only be extracted by spending the energy. So, water potential actually expresses how much energy you would need to expend to pull that water out from the soil sample. It is a differential property, that is there must be a reference, and generally the reference that is given is of the pure water. The water potential of pure water is taken to be 0. Water potential in the environment is always almost less than 0. Why? Because you have to add energy to get the water out. Water potential is denoted by Greek letter psi and expressed in the units of pressure like Pascal, megapascal, kilopascal, and the potential of pure water at standard temperature and without any pressure is taken to be 0 by convention. However, the pure water contains plenty of potential energy. It is not so that the pure water does not have any energy, but this energy is ignored. As I just said that water potential is a differential term. So, for the purpose of reference, the potential energy of water, pure water, is taken to be 0 by convention. So, water potential values for plant water in roots, stems and leaves are therefore expressed in relation to water potential of pure water. I am repeating this again and again that it is to be taken in reference of something. Water potential depends upon the solute, pressure, gravity and matrix effects. So, the water potential of a system can be denoted by some of all these, where psi s is the solute potential, then psi p pressure potential, psi g gravity potential and psi m is the matrix potential. Solute potential, as we had just talked, what happens? Suppose that there are water molecules in a system. Now, in this system, these water molecules, they are free to move around. So, they have a certain amount of energy. What happens when solutes are added? These solutes may bind to water molecules. Now, the movement of water molecules is somewhat restricted. That means, the water potential is lowered. Solid molecules can dissolve in water because water molecules can bind to them via hydrogen bonds. The energy in the hydrogen bonds between solute molecules and water is no longer available to do the work in the system because it is tied up in the bond. In other words, the amount of available potential energy is reduced when solutes are added to an aqueous system. The more the solute is added, it will further decrease. Talking about osmotic potential, it is same as the solute potential. Actually, what happens? the solute potential decreases with increasing solute concentration, and this will cause a decrease in the total water potential of the system. Now, the internal water potential of a plant cell is more negative than pure water because of cytoplasm's high solute content. The cell has many solutes, so the more the amount of solute is there, the lesser will be the water potential. So, obviously, inside a cell, the solute potential is lowering the total water potential of the cell because of presence of solute, which is not there in the outside of the membrane, which is in the pure water. Here in this example, because of this difference in water potential, water will move from soil to the plant's roots cell via the process of osmosis. This is why solute potential is sometimes called osmotic potential. Suppose this is a cell and the cell has lot of solutes. Because of the solutes, here there is low water potential, but outside the water potential is high. The water will move from the region of high potential to low potential. Because of this movement of water inside the cell, a pressure will be applied, and since this is causing osmosis, so the solute potential is also known as osmotic potential. Pressure potential. Pressure also plays a very important role inside the plant cells walls. Plant cells have pressure potential because of the cell walls are differentially permeable, allowing water to pass, but not many solute ions which are too large to diffuse through the cell membrane. The higher solute concentration inside the cell causes water to flow in or be retained. This causes a pressure, that is the Turgor pressure on the walls. Let us come back to this same example. So, what is happening? The water is moving in. Now, the cell is becoming turgid. It is exerting pressure on the walls. This pressure is known as Turgor pressure. Pressure potentials have always positive signs. So, solute potentials were negative, but pressure potentials are positive. And Turgor pressure is very important because it stiffens the flexible cell walls and enables the plants to stand upright instead of lying down flat on the ground. It also causes young cells whose walls are not yet rigid to expand, which is how the growth occurs. Wilting is an indication that Turgor pressure has dropped to a level that threatens the life of a plant. So, Turgor pressure is very important. It is playing a role in keeping the plant upright. It is causing the growth in the young cells, and it is also leading to expansion of the cells. Further, if Turgor pressure is not there, there will be wilting, and wilting can be threatening to the life of the plant also. So, let us see the effect of solute potential and pressure potential. This is a U tube and it carries water separated by a semi-permeable membrane. Now, if you add solutes to one side, right side suppose, what will happen? The water potential of right side will be lowered, and here comparatively it will be higher. So, water will move from high water potential to low water potential. Similarly, if positive pressure is applied, what will happen? The pressure potential will increase. So, here high water potential is there or low water potential is here, water will move from high to low. However, if a negative pressure is applied, what is negative pressure? Suction or pulling force. So, if a negative pressure is applied, it will make the pressure negative, pressure potential will become negative. So, the overall total water potential of the system will be lowered. So, if negative pressure is applied here on the left side, the right side will have more water potential, and now the water will move from right to left side. This is how it works. The next two things that control the water potential are gravitational potential and MATRIC potential. Since gravitational potential is dependent directly on the height, but its value is very small, so it is generally neglected. But MATRIC potential, it is negative. As water adheres to cell walls, it lowers its potential. The walls of xylem are made up of carbohydrates which attract the water molecules. In a sense, matrix potential is equivalent to solute, but it occurs when the molecules are bound rather than in solution. So, this was about the water potential. What we need to remember is that for the sake of simplicity, we will ignore the matrix potential and gravitational potential for a while, and we will talk about only the solute potential. So, water potential of a system, that is psi total, is the sum of solute potential and the pressure potential. This is one thing that we have to remember. Secondly, because of addition of solutes, the solute potential is lowered. So, that means solute potential will have a negative effect. It will decrease the total water potential of a system. In other words, whenever the solutes are added, the total water potential of the system is decreased. The pressure potential, psi p, it is generally positive. So, keep in mind, whenever solutes are added, water potential will decrease, but when pressure is applied, water potential will increase, and the balance between the two controls the total water potential of a cell or a system. Further, I will say once again that the water potential of pure water is 0. That means, when solute is added to it, it will further lower down. That means, the water potential of a solution with solutes will always be negative, less than 0. That means, it will always be negative. So, water potential of pure water is 0, and whenever the solution is there, that means solutes are present. That means, the water potential will have a negative value. This was about the water potential. Now, I have some questions for you to solve. These questions have appeared in previous years in some competitive exams. Let us try to solve a few of them, and then I will give you some as your homework also to try them on your own. Okay. So, first question is, turgidity of cells is maintained by, the four options are osmotic pressure, wall pressure, Turgor pressure and diffusion pressure. Oh, this one is easy, we just talked about it. The answer is the Turgor pressure. Moving on, this is important. Study the following table showing the components of a water potential in closely arranged mesophyll cells, namely A, B and C. So, there are 3 cells A, B and C, and their osmotic potential, that is the solute potential, and pressure potentials have been given. Identify 2 of the following which show correct direction of water movement between 2 cells. Now, since these cells are in contact, water might move from one to other. You have to find what will the direction of movement of water from A to B, B to C, whichever is the answer. So, how will you solve these? What you will do? A, B and C. Osmotic potential is minus 0.21. Pressure potential is 0.05. That means total potential, total water potential is the sum of the two. So, you need to add it and the answer is 0.16. Similarly, for second 0.22 and 0.02, total potential will be minus 0.20, and thirdly minus 0.23. It is simple subtraction, 0.05 and minus 0.18. So, we can see here that the highest potential is here, right, because of the negative value, and the lowest is here. right. So, definitely water will move from A. Water can move from A to B. It can also move from A to C. Water can also move from C to D. What is not possible is movement of water from B to any other cell. So, what should be the answer? The correct pair is third, 1 and 4, that is A to B and C to B. Okay. This is similar question. Two cells A and B are contagious. Cell A has osmotic pressure of 10 atmosphere, turgor pressure at 7 and diffusion pressure deficit at 3 atmospheres. Now, cell B has osmotic pressure 8 atmosphere, turgor pressure 3 atmosphere and diffusion pressure deficit at 5 atmosphere. And what will be the result of movement of water, whether there will be no movement of water or movement from B to A to B, or an equilibrium between the two. Well, in order to do this question, what you are going to do, you will add the osmotic pressure cell A. I will write only for cell A. I want you to solve this question with me. So, for cell A, the osmotic pressure is minus 10. Why minus 10? We have talked that osmotic potential will always have a negative value. So, atmospheric, the units need not to be written here, we are doing the rough work. So, the atmospheric pressure, osmotic pressure is at minus 10, turgor pressure, that is the resultant of pressure potential which needs to be positive, it is at 7, and the diffusion pressure deficit, since it is a deficit, it should have a negative value. So, when we add all these, we get minus 6, and similarly you will calculate for cell B also, and you will see that the movement is from A to B. A has higher water potential and B has lower water potential. So, the movement is from A to B. This is an easy question, no calculations required. Steroid hormones easily pass through the plasma membrane by simple diffusion. We had talked about this, that some substances can move through simple diffusion through the plasma membrane. Why? Because they enter through pores or contain carbon and hydrogen, or are they water soluble or lipid soluble? Obviously, they should be lipid soluble. Moving on, when the concentration of soil solutes is low, the absorption of water is remain normal, decrease, increase or stop? The answer is it will increase. Now, there are some questions for you to solve. In this question, there are 3 cells. You have to identify which one is root cell, mesophyll cell and the cortical cell. Similar osmotic potential and pressure potential values are given. Which one of the following denotes water potential of the mesophyll cell in wilted condition? So, the value of osmotic potential should be higher, equal value of pressure potential, equal to the value of osmotic potential or equal to 0. Then there is a match the column. You have to match whether these values will have a positive, negative or a 0 value. This is water potential of 10% salt solution, pressure potential in a normal cell, in a plasmolyzed cell and matrix potential. Next question, water will be absorbed by root hair when concentration of salts is high or concentration of solutes in the cell sap is high or plant is rapidly respiring. Our last option is they are separated from soil by a permeable membrane. This is the last question. Assertion is that potassium ion accumulation found in Nigella depends on respiratory activity, and the reason assigned to this assertion is absorption of all ions in plants completely depend on the usage of metabolic energy. Choose the correct option, whether A and R both are true or not, whether one of them is true or whether R is the proper reason of A or not. This was all about water potential and active and passive transport. We will meet in next class to discuss more. Thank you.